Christine McGowan. The date is April 6th, 2023. So Giorgio Bernardi wrote in his review article in 2019 that the non-coding DNA sequences in the genome are not junk DNA like was the common thought process behind this extra DNA, that it didn't have a function. But he proposed that it plays a crucial role in regulating gene expression by influencing the structure of the chromatin. Chromatin is the DNA. Proteins and RNA make up all of the chromosomes in eukaryotic cells. If you change the structure of the chromatin, it's going to affect the accessibility of DNA to be transcribed and thus have a really large effect on the impact of gene expression. Bernardi argued that the distribution of this non-coding DNA isn't random, but follows a specific pattern. He said that it's thought of as a genomic code, and that this code is pervasive throughout the genome. His hypothesis is supported by several lines of evidence, including the observation that non-coding DNA is highly conserved across evolutionary time, suggesting that they have an important function. He also pointed out that the pattern of non-coding DNA sequences in the genome is similar across different species, where they're supporting the idea that it's a conserved function. It provides a new perspective on the function of non-coding DNA and their role in regulating gene expression through the modulation of chromatin structure. This non-coding DNA mystery refers to the fact that a significant portion of the human genome consists of this non-coding DNA sequence. For a long time, these sequences were thought to be junk DNA with no apparent function. However, this recent research that he's talking about reveals that these non-coding sequences play a role in regulating gene expression, but also in other cellular processes. In this article, he proposed, again, that the non-coding DNA plays a role in shaping the chromatin. Personally, I find Bernardi's hypothesis to be intriguing and thought-provoking. The idea that the non-coding DNA sequences in the genome play an active role in regulating gene expression and chromatin structure challenges the previous assumption about the nature of junk DNA. If his hypothesis is correct, it has significant implications for our understanding of the genome and its role in regulating cellular processes. I believe that the review article represents a significant advancement in our understanding of the non-coding DNA mystery and chromatin structure. It highlights the need for further research to fully understand this complex interaction between non-coding DNA sequences, chromatin structure, and gene expression.